So please do shout out. And take it from there. Right. So there we go. So the Clinical Practice Research Datalink, we are, or CPRD, we are a government research service. We are housed within the Medicine and Healthcare Products Regulatory Agency with support from the National Institute of Health and Care Research and the Department of Health and Social Care. So we're very much grounded in government. Our remit is to support public health benefiting research. So we don't really limit the types of research you do, provided it has a public benefit aspect to it. And our services are underpinned by longitudinal databases based on electronic healthcare records, predominantly primary care. We've been collecting data for, I think, 38 years. It's our 40th birthday in 2027, so we're planning some celebrations for that. So the data just grows. We never get rid of data unless people opt out. We don't have a statutory function to collect data, so it's very much a GP opt-in model, and then patients can opt out. So if your GP has opted in, you will know about it because they should put it in posters in the practice and inform their patients. And we are a UK-wide service, so the MHRA is the UK-wide medicines regulator. And so when we talk about population coverage, we mean across the UK and have data from all 4 of the developed nations. Our database is approximately 65 million patients, but predominantly, the majority of those patients are either transferred to other practices from whom we don't collect data, or they're deceased. And we have about 19 million patients who are currently active, registered at a GP, for whom we can help recruit into clinical trials. The data we get comes every day. So I go to my GP this afternoon. Tonight it's processed at the software supplier's data center, and tomorrow we can collect that data and then integrate it into our operational systems. So there's usually a 24, 48-hour lag between an event and the data being held by CPRD. But we don't typically make that available that timely. We process the data, we perform quality checks, and we assess its quality for research. So every quarter we do a static snapshot, and we give the metrics of that, a quality assessment, a DOI, and that's the database we suggest people use for research, even though there might be a 3-month lag. But for those that are interested in recruiting patients into trials and you're happy to forgo some of the quality, but for timeliness, we can run searches on a daily, weekly basis to help you find the most appropriate patients for your studies. I say we've been around for a while. We are a UK government service, but we are global in use. That's global with other regulators, global with other academic institutes and pharmaceutical industry. And we can attribute at least 3,500 publications to based on CPRD data, or has used CPRD data. And one of the checks we consistently do is, as we don't have full population coverage, but it is a representative sample of the UK, we continuously check the data against, for example, the ONS census to make sure that it's representative about the UK population as a whole, based on ethnicity, biological sex, and age bandings. Right. As we collect data from GP suppliers, there are 3 in the UK. They have a lion's share. There's Vision, although they've just been purchased by a company called One Advanced. There's EMIS, who've just been purchased by Optum, so those names will change, and TPP. We make the data available. We have TPP data. It's currently being assessed and characterized before we release it. But those are the 2 databases we have available, and we also perform, we map those to a common data model to OLMOP. For those that are familiar with that, it's used particularly across Europe, and the European Medicines Agency are big users of OLMOP. We don't collect free text. So one of the reasons, part of our ethics approval is we can collect data that is pseudonymized at source and effectively anonymized when we make it available for research. So we don't have free text, we don't have names, we don't have addresses, we don't even collect full date of birth month and year is good enough for the types of work we do. But we do collect the drug exposures, the vaccines through DM&D, diagnoses and tests, and referrals via SNOMED codes. Referrals is interesting. We know that referral is made because there is a SNOMED code for that, but then we don't know what happens when you go to a specialist clinic, and we don't have the free text letter that comes back from the clinic. So we do work with NHS England to link to about 17 different data sets. They're not as timely as our primary care data. These come yearly or twice yearly, but we link to admitted patient cares, outpatient data, A&E data, digital imaging diagnostic data, but not the images. So this is purely the coded data. When we get the digital imaging diagnostics, it's the assessment of, say, the X-ray. So if the assessment is a broken or fractured shoulder on the left, that's the codes we get. We don't get the image back. And I tell you all this because this influences the decisions we make when we start looking at generating synthetic data. And we're using a range of studies. The types of studies is not limited. The key driver is it has to benefit the public. So it ranges from epidemiology to look at the incidence and prevalence of diseases, mortality rates, through to the cost of healthcare in the NHS. And we're seeing more and more interest in the machine learning aspect. And this was what drove us to work with our regulatory colleagues on the synthetic data. So that gives you a background of CPRD and hopefully gives you sort of the basis on where we built the work. So is everyone okay? Yeah. Right, so synthetic data, and what was our interest in this? So first of all, so we're all on the same page. When we think about synthetic data at CPRD, we mean completely 100% artificial, generated by some underlying model. We have seen approaches where you take real data and you try and aggregate it or shave off some of the outliers, but so that's manipulating real data. We mean you take a model, and then from that, you can make as much data as you like using some algorithm. There's also this notion of low fidelity through to high fidelity, and it's sort of like a sliding scale. For us, we think of low fidelity as it's data that matches the structure of a data set. So you take a database schema, but that's about it. Everything else in it has no real clinical plausibility, or you can't infer anything from it. But it has its uses, and we've used it within CPRD where people were, before they received the real data, they wanted to understand the structure so they could build up their databases and pipelines. So we would randomly generate values for the fields. Completely meaningless. You would have people who would die 6-7 times over because that was the random SNOMED code, but good enough to sort of build your database and your pipelines. But what we're interested in for this work was high fidelity, and for that we mean data that is capturing the complex relationships between the fields and the relationships between them to the point where it would be completely indistinguishable from our real data that we hold within our primary care and linked databases. So that was sort of what we're aiming for. And our motivation to do that came from the regulatory side. CPRD sits within the medicines regulator, but we don't have a statutory function like the regulatory side. We have a service that supports research. And part of the MHRA's remit is, as well as drugs and medicines and vaccines, is we regulate medical devices, and as more and more algorithms are being put into devices to help make clinical decisions, that becomes a medical device and therefore needs to be regulated along with all the other devices that are available on the market. So that was our real driver for this. And we worked with them. We have the data, they have the regulatory know-how. Can we bring that together to help support the regulation of devices? And we've been doing this for, about 7 years now, but it's been a long journey, but I think we're in a really good place with the data that we're now generating and what we've learned. And there was several motivations, some of which we're still working on, but the first was, can we benchmark these algorithms that typically, we don't have access to proprietary information, but devices come to us, and we need to somehow level the playing field. So if we can control and we know what data goes in, and we're confident that it represents the typical use case, or we can control every test environment bar the algorithm, then perhaps we can help benchmark different algorithms or devices. And that way we can see which ones work best in which scenario or situation with certain types of patients, and which ones are less useful. There's boosting, so that is where you take the real data, but it's really hard to get good data sets with real patients that's quite broad for every use case. So the idea with boosting is if you can understand the relationships with your small data set, can you scale it up? So you have, I don't know, 20 people in a clinical trial, but if you understand the relationships between the data you capture, can you actually create synthetic data for 100 patients? And that way, when you pass that through to validate your algorithms, it gives you more confidence in the outputs that you get. Boosting for us is really scaling up. We don't try and change the makeup of the data, but there are times when you might want to do that. So edge case testing and filling in the gaps. With the edge cases, we would be interested in control conditions, especially clinical trials. You're really looking for patients where they have one condition and that's the one you want to treat with your device or your medicine. But in reality, in the real world, people will have lots of different conditions and lots of different medicines. So actually, could you, and you can't account for that in test environments, it would just be too difficult. But if you could understand and model your patient sets, could you then create the extreme edge cases? And that way, that would then give us confidence of what would happen in the real world when these devices are going to be presented with patients who are, have all these multiple diagnoses and or multiple medicines. And related to that, sort of filling the gaps, where sometimes it's difficult to get the range of patients you need in the right volume. So actually, if you have, looking at this room, a lot of 18 to 30-year-olds, but very few people over 40, could you then scale up the 40-year-olds? Well, because you don't need to scale up your younger population because you already have that data, so you're sort of filling in the gaps a little bit. And then, yes, facilitating data access. So one of the first thoughts was this could be a really good way to help make the access to data more widely available, although that has its challenges, which I will come to later. So that wasn't quite as easy. So that was sort of our motivation. The question is, how do you do it? So there's lots of approaches you can use to generate data. You can take a mathematical model, you look at your values, you work out the means and the variances and the distributions, and then, for every one of those, you can try and populate some values. Or you can use deep learning approaches, such as GANs and Bayesian networks. Spoiler for the next slide, we went with the Bayesian network approach because we only have the coded fields, and Bayesian networks are really good at understanding or capturing that relationship between the fields and the probability of, if you have X and Y, then the probability you will have Z is this. And because we don't have the text, the images, there's no audio, we don't really need to build those complex neural networks. And the other thing that was of interest was transparency. So, if you don't know how the algorithm's built. So if you don't know how the data is generated that you then put in to validate the algorithm, there's a lot of unknowns. But if you can generate data and have a good sense of, is it making plausible decisions on how it's sampling the data, and does it look clinically right as a model is assessed, then you have more confidence in the data that you put in to validate the models, and therefore more confidence in the outputs. What I mean by that transparency is you can pull out the graph, the Bayesian network. So bit fuzzy, but this is trained on cardiovascular data using our Bayesian network. So we took the real data, we did the network analysis, and then we exported learned relationships and patterns so that we could see that it is actually picking up things that are plausible. So if you look in the bottom middle, impot, that's impotence, and there's a strong relationship between male gender and impotence, but that's what one would expect. There's a strong relationship between smoking and impotence, and that's feasible as well. But it also teased out other relationships. So at first glance, you think, is that right? But then when you dig some deep digging, you realize, yeah, that makes sense. So there's a relationship between region and impotence. But the reason for that, when you do some digging, is smoking isn't a uniform activity across the UK, for want of a better word. It's more prevalent in some regions than others. So as region was part of the data set, there is this sort of byproducts that smoking is related to impotence, but smoking is related to region, and therefore, by association, there is an association between region and impotence. So you can look at that, you can challenge it, you can query whether that's right, but at least with the Bayesian network approach, and you pull out the graphs, you can give yourself that assurance that it's understood the data set, it's pulled out the key relationships, and that gives you that confidence that the data you generate next is of high quality. Now, we didn't just rely on the map to settle with Bayesian networks. We did test it with some generative adversarial networks. There was a tool called Gretel. I believe it's been purchased by NVIDIA now, but before it was open to use. And we took some real data, so from our primary care database, and extracted patients that had signs of the fields associated with cardiovascular disease. And then we tried to use that to generate synthetic data using GANs and our Bayesian network approach. And the data sets got progressively bigger. So we start with 50 patients, 100, 1,000, right up to 300,000 patients from our database related to cardiovascular disease. And in every size, the Bayesian networks outperformed the GANs. So this was measured using, it's an open source library called SD metrics, I believe. And what that does, it takes two data sets and it tries to look at compare the values and work out the shape of the data sets, and then does a comparison. So if it's 100%, it's practically identical. If it's 0, they're completely separate. So here we've got, for the 50 patient samples, about 81% similarity according to SD metrics for the Bayesian network. What was interesting as well is with this, as you go through to higher volumes of patients, we started to see with the GANs, mode collapse beyond 200,000 patients, where actually the similarity started to degrade, whereas with the Bayesian network approach, it pretty much stabilized and just flatlined at around 95%. And then from that, we pulled out the graphs for each field, and as you can see, the distributions were quite good, especially with the systolic blood pressure in the bottom left there. I think that's the cholesterol on the bottom right. Ages was slightly skewed, but the distributions were relatively pleased with. So that was our sort of first foray into this. Does it look about right? But then, being in the regulator, we're lucky we have ready access to experts. So we asked some of our clinicians if they could differentiate between our synthetic and our real data sets. So we had 50 real, 50 synthetic, mixed them up. And then we asked our experts to say whether they thought the data was an actual clinical record from our primary care database or one that we had synthesized. With the real data, they typically classified it correctly. So expert 1 got 13 out of 14 correct. But with the synthetic data, that was consistently misclassified as being real. So again, further evidence for us that part of our remit was, can we do this at various data set sizes? And then can we make the data indistinguishable from the real data? And this sort of gave us the confidence that we were on the right track. So that's fine, generate data, but you have to do something with it. Otherwise it's, just a fun activity. So being in the regulator, and the driver with this was ML, machine learning algorithms. The first question was, can we use it to validate machine learning algorithms? And the answer was yes. I can send the links around to these papers later. They've got full details of the methodology and what was done. But we took, I think it was 800 patients from the database with cardiovascular disease and created it with or without. So it was a classifier that we wanted to build to predict risk. And then we took 6 different machine learning classifiers, trained it on the real data, put through the synthetic equivalent, and the difference was minimal between the real data and the synthetic data when validating the models, to the point where I think it was significantly indistinguishable. So then we thought, well, let's reverse it, because this was better than we expected. So can you train an algorithm on synthetic data and then use the real data? So we worked with a company that creates devices to capture blood glucose in pregnant women, and they predict gestational diabetes. So we took some of their data that they had up to, say, today, and then we synthesized it and we trained a classifier. Then we waited 6 months because their devices were still collecting data. We took the real data and used that to validate the prediction algorithms. And we got very similar results to the first experiment where we trained it on the real data and validated it with the synthetic data. So yes, very happy with the results there. So yes, we could use it for ML algorithms, which is one of the driving reasons for doing this, especially the devices regulatory side of the agency. But then we wanted to go further. So can you scale up the data? That was one of our other use cases. So clinical trials, really hard to get lots of data. So can you take a small data set, scale it up, and it still be useful? So for this, we took some liver disease data set, I believe, about 580 patients in the data set. We scaled it up to 5,800 patients, and there was no duplicates. And the correlation between the two data sets, as in the proportion of patients with liver disease and without, remained consistent. And we got similar results when we did the ML validation experiments again with the scaled up data. So I think, but this was scaling up the data as is. So any biases, any underrepresentation, any errors in your raw data, we would magnify it. So if you had missed something, that wouldn't be in the final data set, or if you classified something in the real data, that will be scaled up tenfold. So this was just, can you take data you're happy with and make it bigger? But that leads to, how do you handle underrepresentation bias? So we worked on a method called Bayes boost. And what Bayes boost tries to do is identify the fields that is underrepresented. So without a board, this will be hard to explain. So you take your data set and then you train it, and then you have a classifier. But then you select which field is what we call the protected characteristic. It can be anything. It can be ethnicity, can be age band, can be region of where you live. And then using that knowledge, it calculates how certain it is, given say an ethnicity, how confident is the output of the prediction based on the data set as a whole. And then if the uncertainty is at a certain threshold, that becomes a candidate to boost that subset of the population. So I'll try and talk through this. This graph was one of our experiments. So the green is the real data, and this is for cardiovascular disease. And we marked ethnicity as our protected characteristic. So this is the thing we felt in the data set may not be representative. And as you can see, we had 22% were white or not stated. A good just under 70% was other ethnic group. And when you look at the other ethnicities, there's very, very few percentage of patients that were marked as those ethnicities. So to test our theory, we overcompensated. So the red is what we simulated, but we added in, we manipulated the data. So in the first category of white or not stated, we made the proportion really small, hence the red bars right down. But for Black Caribbean, we pushed the number of patients as a proportion higher. And then when you run the uncertainty analysis, so you train the model, and then it says, okay, given this is the model, if I was to take the white or not stated on the biased data, how confident am I that I'm predicting cardiovascular disease correctly? And the answer here is not very certain, just under 30% uncertainty. And so that becomes the candidate then for boosting it, because the general rule was if the percentage of uncertainty is higher than the percentage of people with your protected characteristic, you need to balance it out. So if you look at the Black Caribbean bar, that's really certain because we overpopulated the Black Caribbean ethnicity in the data set. The uncertainty measure is practically zero because it's very confident that given that characteristic, it is making the right prediction. And then the blue bar is then a synthetic data set that takes that knowledge and tries to readjust the proportions of the patients so that you have a more balanced data set. So as you can see, the two columns where the uncertainty was high now have a higher proportion of other ethnic group patients and white not stated, whereas the proportion of the other groups has gone down slightly, but not enough that it goes below the uncertainty threshold. So you try and manipulate the data set with the synthetic generation to come up with a more balanced approach. And you can pick whatever field you like as your protected characteristic. So in this one, very similar, well, this was smoking. So I think the categories are 0, non-smoker, X, light, medium, heavy. I don't know what classifies as a heavy smoker, but anyway, heavy is 4. And then we reversed it. And then the sort of top right is the uncertain classifier. So because this was based on our biased data, where we've reversed it, it's very uncertain in its predictions for non-smokers, very certain in its predictions for heavy smokers. And then in the bottom left, that is deboost. This is the synthetic data set that has taken that into account. And as you can see, it's skewed some of the proportions back towards the non-smokers because it's tried to create a data set where it has enough in each pocket to make a sensible or a better chance of making the right prediction. This approach is slightly different from some others, although I believe smoke has changed now. Whereas with smoke and adaptive synthetic sampling, what they do is if there's some uncertainty or it thinks there's not enough population in the data set, it adds more and more, but it's not done as a proportion. So if you look, that's why in the smoke and the ADA-SYN data set, you go right up to 60,000 patients. It's not tried to adapt the shape of your data set, but it's going to just add more and more data in the hope that you then have enough to create a classifier that's works and is confident. So that's how our approach is slightly different. We don't add more data, we adjust the patients in the data based on what characteristic you have chosen. But the conclusion was, yes, you can sort of manipulate and adapt the data to try and accommodate for bias or underrepresentativeness. Right, little split. Any questions thus far? Or are you happy for me to carry on?
I've got a couple of questions, but I'll wait to the end.
No worries. Right. So emulate. So we've got these methods, but what we wanted to do is our remit is to support research. So can we take this and put it behind a platform where people who don't know about synthetic data, but do know about or they might know about synthetic data, but really they want to get their hands on the data to do the interesting stuff like the machine learning algorithms that make their devices. So we built a platform, and our driving motivation for this was no code. You can use it without knowing how to code in Python or R, and you will get something sensible at the end that you can then use in your research. So whether that be, validating algorithms or developing new ones or with clinical trials. We don't want to own your data, so you own all the IP. We don't even keep the model. We destroy it once you've downloaded it because it's yours. We don't want to profiteer off the back of everyone else's data. And what you get is, at the end, a nice report and your synthetic data. And we've condensed it into 3 steps. So I'm not going to do a live demo. I was tempted, but I thought that would be, tempting fate to do that. So step 1, you upload your data. At the minute, we limit it to 200 megabytes, but that's more of a resourcing issue on our cloud environment than a technical limitation. Well, that's it. Choose your file, set some parameters, and at the minute there's 3. So the first one is you tell it if you have a primary key in the database, because then it ignores it, or it doesn't try and look at distributions and model primary keys. It knows that, okay, for every line of data, it needs a unique number because it's a primary key. So you just can tell it, ignore that. We've set it, the increased data size can be like for like. So 50 patients go in, 50 patients come out, or you can go up to double. Again, that's more of a resource constraint than a technical one. And then we're looking at noise. I'll come on to that because that's more further work, which is if you set that to 0, you get high fidelity, the data as accurate as the data that goes in that we can make. If you set the noise up to max or higher, it will get rid of some outliers. It will inject some random values. It's designed as a privacy approach, but we're still working on that. But that's the general premise. Review, and then you're at the point of no return, and you get your data. And what you get is, I won't show you the data. It looks like a CSV file, so not terribly exciting. But then you get a report, and for every variable in your data, it will show you its analysis and produce a chart to show you how similar the synthetic is to the real data that you put in. So in this, what have we got? 15,000 patients went in, 15,000 came out, and 32 columns each. So that's the platform. Trying to make it as simple as possible. And yes.
The values in the data set, are they numbers or can they be categorical values?
Categorical numbers. It's good on numbers and categories that you map to a number. So if you are very simple categories. So if you look at the marital status, I think is 0 through 3. The sex will be biological sex from the database, and that can be M, F, or U for unknown, I think. So very simple categories. Yeah. It does.
You need to specify that in advance what the categories are.
It looks at the values and uses that to infer what data type it is. So there's this work. The more we look at this work we can be doing on that. So specifying the actual data types and ranges is something we could do, but it makes the interface really complex. But to start with, we wanted something just really simple, and behind the scenes, we calculate whether, is this an integer? Yes, looks like it. What's the range? We'll assume that's the range. Are these characters? It's not why though. We specified the primary key because what we don't want is you have 15,000 patients. You start, in ideal world, it starts at 1 and goes 15,000. But if you pull them from the database and they're random numbers, and it starts to think that's some sort of distribution, you may end up with duplicates. But if you say this is the primary key, doesn't worry about it. It just knows every row must have a unique number somehow. That's meaningless. Yes, so that's. But yes, it's all sorts of work that's going on, which now leads me neatly to that. Right. So have that sort of operational, kind of. It's not publicly available yet, but it's getting there. So the first thing is some work that one of my team, they're doing a part-time PhD on this, is can you use our data in clinical trials? So what Barbara is looking at is if you take a full clinical trial data set, but you have, maybe limited number of patients that participated, can you synthesize that, scale it up, maybe accommodate for some unrepresentation within the data? And would that still give you a valid clinical trials data set that you can then pass on to perhaps the regulator itself as part of your submission? So that's still ongoing. But or can you create a control arm that's entirely synthetic? So you have your two sets of patients, but the real patients is in the clinical setting, and we take the control arm and synthesize it. Some advantages that you can reduce the number of patients you need to recruit, because that's always a real challenge in clinical trials. But also, if you're having difficulty to make sure you've got the representation you want in your clinical trial, perhaps the boosting can help with that as you get to the end of it. So that work's ongoing. I think as this comes more towards conclusion, I'm sure Barbara would be happy to talk to you in depth about that. Just sort of related to that is, because we sometimes get asked about in silico trials. We're not doing in silico trials here. So with in silico trials, you take your data and then you model what would happen, and you simulate it in your virtual environment by changing your interventions or changing certain aspects. We're not doing that. It's purely, we have some data and we can synthesize it and it looks like the real data. But that can be used for in clinical trials. If you want your base patients, then, before you start modeling what may or may not happen, you perhaps could generate your synthetic population at the start, and that would then lead into the in silico trial work. And then that sort of leads on to some of the other questions we get about digital twins. For us, we're not doing digital twins. That has a connotation that there is a real person and we know who it is, and we've synthesized their sort of medical records, and then we're sort of, with their digital twin, making interventions or decisions to see what the output is. But we're not doing that, and we can't do that because we actually don't know who our patients are. There's, as I said earlier, it's pseudonymized at source. It's effectively anonymized when we release the data for research. So one, we don't know whom the twin would be, and then secondly, that would have some, it would go against our ethics approval, because yes.
It's a twin on a population level.
It's a twin on population level, yeah. But but I think when people talk about digital twins, it's like there is the physical real Darren, and then I've got all my records. I've synthesized my records, and I can prod and do things to my digital Darren. But you're right. It's a digital population twin at a high level. Yeah. Privacy enhancing technology. This is potentially, yes. I think this is where technology needs to work with the information commissioner and lawyers and information governance experts. I think it's a real complex, knotty issue with lots of factors. So with our we thought our synthetic data, when we started this 7 years ago, we could make that more easily available, but it's not the case. There's some real challenges with that. So until we can come up with a definitive framework of what needs to happen, at the minute, our synthetic data is treated like our real data. So you have to request it with a real research proposal. It has to be destroyed after so many years, and you have to tell us you've destroyed it and all the rest of it. The reason for that is, I think it comes down to, lots of things. As I said before, the low fidelity data where you take a schema and you randomly fill it, that's private because, that has super privacy. It has very little use though, but you would never find a real patient in there. But there have been attempts where you take a model and you somehow reverse the model to go back to the real patients. Then there's a question of, if that was to happen and you got back to the real patients, your data isn't anonymous anymore. So I think what needs to happen is lots of us need to get together and somehow work out a framework for, if you've generated your data doing this approach and it's on this scale and this was the source data, and you're not using any real data as part of the synthetic process, then perhaps that's low risk and we can relax how we make it available and have some sort of framework or threshold. I'm not a legal expert, but I know the IG around this and the ICO is quite keen to look at this. So that's why I put potentially yes, but it definitely needs a lot of thought, and a lot of thought beyond the technical side also. Although one of my staff is looking at this issue of privacy versus utility as a part-time PhD, and the premise is, although I think the premise is, if you have a data set, synthetic or otherwise, there is this tension. You can have really, really useful high utility, but that comes at the cost of it's not very private. And the more you make it private and obscure it, the less useful it is. And so you end up with this sort of tension. And so in an ideal world, you'd have both. You want something that's completely private and has maximum utility and everyone's happy. But in the real world, what will probably be is this sort of trade-off where you reach the point where it has the utility that you need, but with the privacy that ensures anonymity for the patients. And there's all sorts of things to look at. She's looking at here, like, is this to do with the size of the data that you can make things more private or not? Is it to do with how do you calculate this trade-off? And then are there methods to do that? And even we're looking at privacy as a data set as a whole, but actually, is it really that there are some parts of the data set that are inherently less private than other parts, in which case you can somehow maybe segment the data if that's the more appropriate use. So again, this is ongoing stuff, and I'm sure Yelenia will be happy to talk in great detail about this, but it comes down to it's not quite a privacy enhancing technology yet. I think there's potential, but there needs to be some more work on that area. And quickly, relational database, our synthesization approach works great with data sets, which is effectively one table, but the reality is that comes out, there's a step before that, and it's a very complex database, and the approach falls over at that scale and that level of inter-table dependencies. So if you take CPID, Aurum is our biggest primary care database, but you have the idea that an observation is made of the patient by a member of staff. Lots of observations grouped together become the problem. An observation may lead to a drug issue. There's lots of relationships and interconnected bits there. So can you synthesize a full relational database with the referential integrity, the primary and foreign keys, and all the rest of it, and still maintain that high fidelity approach? I don't really have an answer for that yet. And then other types of synthetic data. So CPID only collects the coded data. Now, depending on who you ask, there's 30% of the value of the data is in the GP notes and the referral letters. I've had quotes of 60%, although I think that might be the volume is probably 60%. You can't compare a single SNOMED code to a huge letter. So, but let's say that, but there is value in the free text, but we can't receive it because it's inherently risky. And our ethics approval and CAG approval is that it must be effectively pseudonymized at source, but we're in a catch-22. We don't know how to anonymize the free text data. We can't collect the free text data because it's not pseudonymized, but we don't know how to pseudonymize it because we can't collect it and look at how we should be able to do it. So is there a role for, say, large language models in this where you can create realistic looking free text from GP notes that then allows us to do things? Either how do you anonymize it, remove the identifier bits, so you go back to the framework before, the utility versus the privacy, you mask so much of the text, it actually has no value. Or There are approaches where you can start seeing if you can map some of the notes to the SNOMED and the DM&D codes. I think that is an approach, although I think there are challenges with that. So if you go to the medical observation table in Aurum, it's a very definitive, yes, this patient has this observation. There's no qualifiers. Whereas in the free text notes, there are things such as it is unlikely that there is a potential of it is not this. So how do you capture those qualifiers if all you're doing is taking the data and trying to map it to a single SNOMED code? So I think you need to capture that subtlety of the qualifiers as well. But I'm not saying it's impossible. I think there's definitely work we could do there. And perhaps, these things are sometimes treated in isolation. We've only worked with coded data because that's what we've got. Some people work with the free text data, but actually they're inherently linked. You won't have a free text field and some associated SNOMED codes from the same consultation that bear no relation to each other. The SNOMED code will be the definitive observation, and the free text will support that clinical decision. So I think bringing them together somehow to make more informed synthetic data. And at that point, and I don't know, this is way out there, but would that then help us with classifiers? We're looking at risk detection with ML algorithms on the coded data, but actually would the free text support that and make better decisions by the algorithm? I don't know. They're just starting to put it out there. And on that, that was a whistle-stop tour of our synthetic data. Thank you very much. And I'll try and answer any questions now.
Thank you.